Hi, I'm Yehlin. I'm a fifth-year grad student working in Sergey at MIT Biology Building, and currently I'm mostly working on protein design, de novo, or antibody engineering, and I'm also working on optimizing the protein properties protein stability and affinity. Uh, yep. Then can I get started?
Yeah, let's go.
Okay, yeah. So today I'm gonna talk about my two recent work of which is protein design tools, the Boltz design and the protein hunter. So it's basically not fine-tuning or training the new models, but inverting the structure prediction models or exploiting the structure hallucination for protein design purposes. Yep. So I think as you already know, deep learning-based filtering method really improves the experimental success rate of designing proteins. So for example, like previously before AlphaFold 2 or like 1 came out, people mostly use a physics-based a filtering method like Rosetta. And then that method usually requires like people to generate 100,000 of designs and then filtering them based on this like a physics-based in silico method and filter them out and experimental ability 1,000 to like several thousand proteins to just get a one binder. But as more of like computational tools are coming out, we can see that like this structure prediction tools like can help to filter out the protein designs, but also the like confidence score from the structure predictions modules are really helpful. Like so now people like develop the like IPSAE and like PAE score, IPTM score, like which are really good confidence scores that can like filter the bad designs like from good designs. So, yep, this deep learning-based filtering and designing method really increases experimental success rate a lot. So for I just like prepared this overview of like general protein design pipelines, and I just like simply categorize into three different pipelines. I definitely think there are like more different method other than this one, but yeah, to like just give a brief overview about this methods, like I just like categorize into three, and I can say the first method is like multi-step gradient updates method. So instead of like retraining the model, like we can just invert the structure prediction model because like we assume that these models like not only learn the prediction, but also learn about the like sequence probability and the like sequence space. So the way it works is like you always like set the like customized optimization functions. So like a contact loss or the confidence lows, and then you are actually making a prediction from the random Gaussian noise, predict the structure, and get the score, and backpropagating that score back into the input sequence instead of like updating the modal weight. So in that case, if you backpropagate multiple times, like now you can get a like a sequence that can like achieve the higher scores. So you repeat this process until you get a like highly optimized scores, and this way is like basically you're co-optimizing the structure and the sequence together. The good thing about this method is like it's really can optimize whatever you want instead of like refine tuning or retraining your models, but you can always like give it as a like customized loss with a like single like the few lines of codes. But the
Sorry,
bad, uh-huh, sorry.
Yellen, now seeing your figure here, the one on the right, C,
Mm-hmm.
um, or yeah, yeah, can we maybe go talk about the figures? So the first one, right, there
Mm-hmm.
we start from a random point, and maybe we even start from 0, 0. I guess it's not entirely the case
Mm-hmm.
that you do. But yeah, you start from some random point in this sequence space, and
Yeah.
now you just for illustration purposes, always put this point to 0, 0. But we
Mm-hmm.
start from a random point and then we optimize. We do our gradient descent and yeah, it's not even stochastic gradient descent, I think, right? In Bindcraft and Boltz design. And then we end up at some minimum and that is our design. In the
Yeah,
other
that's
one, we sample the distribution, like in RF diffusion.
mm-hmm.
And then in Protein Hunter,
Yeah.
there the visualization is a bit different here because you're only showing a single trajectory or a single generation instead of showing multiple generations, right?
Mm-hmm. Yeah, the
Yeah,
reason we
okay.
show this is
S-
like oh, sorry, go ahead.
So, and then why do you put it into the modes? Because you would also end up somewhere else,
Hmm.
no?
Yeah,
Not in a
that's true.
mode.
Yeah, that's a possibility. But here, the reason we just draw this like one trajectory is like instead of like totally restart the whole process like from the random seed, we can like actually redesign the existing design. So like we want to emphasize that our process is now like drawing the random seeds and like predict a bunch of structure and filtering based on that. But instead of doing that, we can start with the one seed and then like maybe branch out from those like already existing the suboptimal
Ah,
solutions. Uh-huh.
okay. So if I tell you, please give me a set of 10 designs,
Mhm.
then will you go ahead and run your model 10 times from like the same starting point or from different starting points? Or will you run it once, make one trajectory, and then give me 10 samples out of that trajectory?
Yep, I think I usually just run 10 different random seeds, but the way I filter out is like, for example, like if you get a like good enough solution at the like first cycle, I just like keep that example as a the final design, and then you also repeat it multiple times, and like you will like definitely get like some
Yeah.
designs like from these like cycles, and you can like yeah just collect a bunch of like data set or a designs that passed the filter that you set up, then like maybe run the validation. Uh-huh.
Okay,
So
but
I
I'm
will.
foreshadowing here maybe a bit too much, and for the benefits of it being a coherent presentation, I should maybe let you continue in your um, layout.
Hmm. Yeah, so I don't mean that like you we can like generate everything with the like single seed. Yeah, but yeah, like I totally understand your point. And like this figure seems like oh we can like do everything with the one random seed, but we just wanted to emphasize that with the random seed, like we can just branch it out like by cyclizing the models and also like get the multiple solution and further improve the suboptimal solution instead of like getting that solution as our final product. Does it make sense? Hmm.
Yes, absolutely. Don't let
Yep.
me derail the presentation as well. Let's
Hmm.
resume.
Yep. Sounds good. Yeah. So Hannah's already briefly explained about this method, so I could like just give about this general overview and yep. So yeah, I want to first talk about the bulk design method. So I think you guys are already familiar with the structure prediction model, like but like to explain about like inverting this model, I'll like first start with explaining about this AlphaFold like model. So for example, so AlphaFold models, like as you already know, it gets a like sequence or multiple sequence alignment as input, and like as an optional, you can get a template as an input, like draw out this model, draw out the MSA module, and the evil former model, you can process the like global information of the sequence and also can get a like contact information like by processed by the histogram, which is showing the probability of the distance between the all residue versus residue. And using this pairwise information as an input, throughout this structure prediction model, you can like predict the 3D all atom like structure information, but also you can get a like confidence score of, for example, the PLDT, which is the side-wise confidence score, and a like PAE score, which is a pairwise confidence score. But after this AlphaFold 2 came out, like some of the people like Sergey and yeah, other folks from EPFL and also yeah, David Baker Lab, they thought about this like thought about the alternative like ways of like using this method. So which is like, can we basically use the structure prediction model for designing purposes? So what they have developed is they thought the to design a protein, all you need is are loss and the confidence metrics. Like because anyway, like to design protein, we need to get a good contact in terms of like intercontact and intra contact within the protein, and we also want to ensure our designs like to have a high confidence scores. So to do this, like what we want to do is like design some sequence that can optimize and like satisfy those score and like contact information. So the the thing people figure out is like, oh, since these structure prediction models are like working with any kind of like sequences, like any random types of the sequences, what if like we initialize this whole process with a random sequence and get some scores and the backpropagate the score using gradient optimization back to this input sequence and repeat this process until like getting a high confidence score. So for example, like maximizing the PLDT score but minimizing PAE score. Uh, and the cool thing about this model is it's not only working with the one-hot encoded sequences, and AlphaFold works with a soft losses input. And this, if you initialize everything with the soft losses, like the overall like optimization process is more stable. And so by like starting with the soft losses, like throughout the whole process, you can push this like losses to be more of like one-hot encoded. So in that case, like you can generate your sequences like using a very a stable optimization process. So starting with the soft losses, you can repeat this multiple process until you get a like highly confidence structure and the sequences. So yeah, the first model that used this whole AlphaFold 2 based hallucination pipeline is Bindcraft. So this Bindcraft is a one-shot design of the functional protein spell binders, and it's really working well, like for many easy but also very hard targets. And uh, the experimental success rate of this pipeline is really high. So a lot of people love this whole pipeline, and they also roughly filter out the designs based on the not only the deep learning based method but also like the Rosetta like physics based method. So the whole process looks like this: initializing everything with the target protein. You initialize the binder, the random sequence for the binder, and then you co-optimize the backbone and the sequence together using AlphaFold 2 multimodal. And once you get a highly confident trajectory, later on you redesign the the backbone sequence of this your binder using a soluble MPN, and then to ensure your designs to be solubilized, but like fixing the interface residues and optimize like more using a like MCMC based method and using an AlphaFold 2 monomer. Like you validate whether this binder bind to this target protein of target. And I thought this is really clever because like AlphaFold 2 monomers are not trained on the PPI data set, so it can really reduce the bias and yeah, like overfitting issue. So based on this filter data set, you also run the Rosetta the relax pipeline and filter them based on the like number of unsaturated hydrogen bond, delta G, and then like the percentage of the hydrophobic interface. Yeah, question.
If you go back one slide,
Uh-huh.
like this is sort of the core of what these procedures are doing.
Yep.
This optimization based on gradients to then
Mm-hmm.
optimize these predicted methods, metrics like PLDVT, PAE. And these are actually even classifications where you
Yep.
right have your or classifications that are combined in a soft manner. And
That's true.
now this is sort of like doing an adversarial, or it is doing
Mhm.
like doing an adversarial attack on your model, right? You notice we have maybe some image or so, and then
Yep.
we it's the image of a cat, and then
Mm-hmm.
we want to change the image such that our model outputs the prediction that it is a dog.
Mm-hmm.
So then we take the logit for, or we maximize the model's prediction probability for it being a dog. And
Mm-hmm.
then we make some pixel changes in our image. These pixel
Mm-hmm.
changes are small, and then when doing these adversarial attacks, what we end up with in the end is actually something that the model is like super sure that it's a
Mm-hmm.
dog. It classifies it as a dog, but
Mm-hmm.
to us humans, it still looks like just a cat. And
Yep.
why? So if you the end of this optimization procedure is not
Mm-hmm.
something successful. It's not something that looks like a dog because you're
That's true.
just exploiting the features, or like you're just exploiting um the the understanding of the model that goes in the direction of making this a dog. And so this is
Yep.
not something that actually makes
Hmm.
something ad hoc.
Yeah,
And
we totally agree.
yeah, then
Mhm.
why does this work for proteins?
Hmm. Yeah, we totally agree. And then, yeah, I don't think like just using the confidence module only for designing optimization is a good way because like you'll like end up getting like bunch of like just adversarial sequences. And I know that like all the AlphaFold 3 child models like their confidence model are not like super accurate enough. Like sometimes they're not really good at like ranking the like designs as well. So I don't really think the confidence module is like perfect way of like scoring your designs. So here, I think the most important like thing is like the uh the most important the loss functions here is the inter and intra contact. So I think those are essentials, and the reason that people added the PLDT and the PAE score is like just ensure at least like your proteins like to filter pass your whole pipeline, and also we assume like there's something maybe like the model like learned throughout this like confidence module. So if you look into their like config file, like most of the weights are like focused on this inter and the intra contact loss, and then like the PLDT and PA component is just really minimal, like maybe like 10% of the contact loss. So what I want to say is like yeah, we mostly like focus on like maximizing and optimizing the context, not the confidence scores. But this is like
Yeah. And
kind of
these contact losses and optimizations, they're kind of local, right?
yeah.
Because you only consider like the minimum or the ones that are already scoring the worst, and these are the ones that you improve.
Yeah, the thing what we wanted to do is make many like contact as possible, but that contact probability to be very like sharp enough. Like so sometimes like if you
Yeah.
look at the histogram, although like you have a the peak at the contact region, but still like those contacts are very broad, which means like the model is not really confident about that contact. So what we wanted to optimize is actually like making the contact to be maximized, but also the contact probability to be very sharp.
Mhm.
Mhm. Yep. So, so this is a background about the previous method. And then I think the both design is really a very similar to Bindcraft method. And yeah, we actually repurposed this method like for all autumn like protein generation. So yeah, after this method came out last year, like in the fall, like lots of like AlphaFold 3 likes model came out, and now those model like can all do the all autumn like protein structure and the sequence modeling. So we thought, oh, it'll be really cool like if we can also utilize the same backpropagation method like Bindcraft, but also design a protein not only toward a protein but also small molecule and RNA, DNA, and model PTM, like so all this kind of a lot of like stuff. So, but the one thing we figure out is the huge difference like between the AlphaFold 2 model and 3 models are now AlphaFold 3 style models have a diffusion model, which usually requires like 200 steps of like prediction during the inference time. So we thought it would be like really expensive to backpropagate through those whole like 200 different steps. So we thought, oh, it's not really possible, and it'll be really time consuming, like to backpropagate this. So the thing we figured out is like since the pair so, oh, before explaining this, this AlphaFold 3 models, like similar to AlphaFold 2, after the pair former, it process all the like pair features of the model and output the histogram, which is like actually showing the global view about the contact information. And given that this pairwise representation and the single representation as the input, the diffusion model like actually denotes the 3D coordinate of the model. But one thing we figured out is like when you make a contact map from the 3D all atom structure, and then when you make a contact like from the histogram, like they looks almost same, like for very confident structure predictions. So for example, like we tested with this like BAT-V1 target and generate a binder, and we got a contact, and then the other contact from the diffusion model, and when we compare that, like most of the intercontacts were matched. So the thing we figure out here is, oh, maybe we don't even need to use the diffusion model like to backpropagate this loss. What if like we just use this paraformer trunk, just like focus on the contact inter and the intra contact loss instead of like ignoring the confidence core and all the like 3D all atom like structures. So, and before doing this process, we validated, oh, like the PIK, the precision of the K contact score between the performer and the structure module, and they're like really highly aligned. So as you can see in the left, the precision of the structure module are like almost linearly correlated with the performer. So they have about the same precision. And as you can see in the right, we also assume, oh, if the diagram have a really high confidence score coming out from the intra and the intercontact, maybe they can also reflect like something on the confidence model. So we also plot the site-wise a confidence score and the intra contact loss, and then we also plot the interchain contact loss and the um, pairwise confidence score, which is the inter-PAE score, and you can also see this linear correlation. So we assume that, oh, it'll be really nice if we can like just maximize the confidence and the contact of the intra-inter contact, and otherwise like PLDT and the PA score will be like followed. So the whole both design one pipeline looks like this. So you give a target. It can be anything like a small molecule, DNA, RNA, or a protein, and initialize the binder sequence with the random Gaussian noise. So you input this model to the performer, and you get the site-wise and pairwise features, get a histogram, get the contact loss, and backpropagate it back to the sequence input sequence until you get a highly confident score. And the second pipeline we figured out is maybe what if like we also add um, the confidence core as our like customized loss. So instead of backpropagating the confidence model score back to diffusion model and the performer module, here we what we have done is like backpropagating confidence model directly to the performer trunk. So instead of like doing this whole process, we can just bypass the diffusion model. So it's much more quicker than like doing the whole process. Does it make sense? So after
Yeah.
we do this whole process, now we get a like finer structure and a sequence. So similar to Bindcraft, we just like fix the interface regions and redesign the rest part of it because like in the previous paper, we have seen if you like totally regenerate everything with the this backpropagation method, like sometimes these proteins are not solvable enough because like you can sample some adversarial sequences. So instead of like fixing all of the residues, we redesigned the backbone part of the proteins using a ligand MPNN and the protein MPNN. So yeah, the summary is yeah, this method is just like backpropagation optimization method, and we have a different like stage. It's really similar to the Minecraft paper. So starting with the very soft losses, the whole process is pushing it to the one defined solution at the end. So starting with the random like gumbo noise, we in the stage two, we move into the softmax stage, and then like in the stage three, we optimize everything in the one hot encoded stage, and then like final stage, based on the like per residue level confidence score, we pick like some residues that have a low confidence score and like randomly mutate those sequences like into other sequences and check the IPT and PLDDC score and decide whether to pass or like stop this designs. So after this whole process, like you will get a like one structure and a one sequence. Yeah, so before like talking about in silico research, we tested it, experimentally tested it, this design in the wet lab. Like so so far we are like working on like a bunch of different targets, but not so far we got a like experimental research like from this PDL1 and IL-7 7 receptor alpha. So for this PDL1, the cool thing is like we designed some beta shed binders, but it actually does not like bind to the into the same binding pose like where this PD1 binds to the PDL1. It has a like a little bit of different like binding a binding mode and a structure, but it still like can inhibit the interface of the PD1 and the PDL1. So we're like still evaluating this, and like this has a like a hundreds of nanomolar KD range. We also and also Adaptive Bio, they offer to like experimental validate some design. So we also designed some binder toward IL-7 7R alpha protein, and we got these two hits like from our 10 submitted sequences. So they're also like 300 nanomolar range KD designs. So now we are like more focusing on like small molecule binder designs and the DNA RNA binder designs in the well lab. So for the in silico success rate, we evaluated on these four different targets, like coming out from the Earth diffusion a lot of papers. So we given this the small molecule, we designed like bunch of different binders and finally, like we made a final prediction with AlphaFold 3 and then measured the success rate based on the PLDT and the PA score. And we confirmed that we could achieve pretty high confidence score. And yeah, we can like even improve the success rate even more like by iterating the whole performer and the confidence module. So yeah, in the summary slide for the Boltz design one is a generalizable tool for like generating binders to wide range of biomolecules. So yeah, we can like give any like target to the model because like we are totally relying on the power of structure prediction model. But the cool thing is you can always customize laws. So if you want to maximize the intercontact, you can just increase the weight for intercontact laws. If you want to also maximize the negative helix loss like to generate more beta shears, you can also like penalize the position I and I+4 in the contact map. So in that case, like you can reduce the like amount of the helix generated from the whole process. And yeah, we always like use different models as a cross validation model like a Taiwan or AlphaFold 3 because we always assume like this model can hallucinate and actually generate or like predict the versatile solutions. So we always like try to check whether this like design structure and then like
Hmm.
predictive structure have a self-consistency between these structures. And finally, yeah, maybe if you want to more strictly filter out your designs, yeah, you can like use some docking tools like Gina, or like for metal, like you can also use this all metal 3D like to track the coordination of your proteins with the metal ion and pi Rosetta or PPI interactions. So I think yeah, this is like really we don't use any fine tuning, so it's really easy to tune and like customize for your own target. So I'm still working on both design to pipeline, and this is like actually just using a Bolt 2, but instead of Bolt 1. And the only difference would be like you can input the template information. So in that case, you can do the motif scaffolding and also like not only giving an MSA as information, but you can like just give a template as an input and reduce dependency on the MSA to remove the evolutionary bias. And you can also do some like hotspot conditioning. So this is oh, I think this animation is not working. So yeah, this was the overview of
Oh,
oh,
ah,
wait.
okay. Sorry for the interruption. It was just there was a
Yeah, this is the
little.
overview about the pulse design. So, oh,
All right. I just think the connection is not super good. But anyway, this is a good point where I wanted
oh,
to ask something anyways, if I may.
mhm,
Okay, good. Just wasn't sure if you're still hearing me. So now the filtering, right? Alpha 3, Chai 1, and orthogonal methods, so to say, then how much do you weigh, or do you have any intuition or any evidence, maybe even on how much you care about the Nina score, or how much I should care about the Nina score versus Pi Rosetta versus all metal?
mhm. Yeah, so oh, you meant like just Nina score or also other
No,
like
all of these,
all of
all the metrics besides AlphaFold 3 and Chai 1.
mm-hmm. Yep. So uh for the AlphaFold 3 Taiwan, for example, like after I got a like IPTM like higher than 0.8, like IPSAE mean like higher than 0.7 and like PLDT like higher than 80. And then I try to use just a default value that I set it up like for the secondary method for also for the Nina. Oh, I just rank the value and maybe like try to like at top K of the of the designs, because like for this one, like it doesn't really have a like the threshold for the specific target. So I just like use a Nina for the ranking process, and for the all metal 3D, you definitely know like for a specific metal, yeah, you need to like make the like the four the tetrahedral or like six coordination numbers. So I try to like check whether our designs like also satisfy that coordination. So it's like just to check the coordination. And then for the Pi Rosetta, I think yeah, Martin mentioned that like Bindcraft with the Bindcraft is really helpful to filter out the designs that have a very low hydrogen bonds and very high like broken hydrogen bonds, which can actually yeah decrease binding specificity. So I'm trying to rely like on the same default setting. Mhm.
Okay, so what are the metrics from Pyrosetta that you use?
Oh, so I use a default from the Minecraft, so I can't remember like whole scores, but like interface hydrogen bond percentage less than 35%, and then unsaturated hydrogen bond less than 3, and then like hydrogen bond higher than 5, something like that. Yeah, but I can't like totally remember
Hmm.
everything. Uh-huh. Maybe I can
I
share
would
this.
be very curious what the wait, isn't Pyrosetta also the thing that you can't use it commercially?
Oh. I'm not sure about that, but I heard that like recently they figure out, yeah, that there's a way to use Pyrosetta commercially. Yeah, so
Well, what we do in Bolzgen is we just implement counting of hydrogen bonds and
mhm.
counting of salt bridges, and we use those, and then we don't rely on any um, external software or packages that have
Got
permission
it.
issues like that. Well,
Hmm.
actually, we use hydride as an external package that does some hydrogen stuff, but that doesn't have any package issues.
Hmm. Got it. Yep. Any more
So I had
questions?
a query
Yeah.
here, where like which downstream sequence designer and refolding pipeline did you use for bold design one results?
Oh, which pipeline?
Yeah,
Oh,
yeah. So like it was the protein MPNN plus AF3 refold versus
mhm.
the bold designs one default, or like you tried something else.
Yeah, default is like for protein. I use a soluble MPNN as a default, and yeah, everything like the cross validation is done by AlphaFold 3. But for the
Uh,
ligand, I use a ligand
mhm.
MPNN. Hmm.
Okay, okay, cool. And for the like the hotspot conditioning template, so you had certain features from the bold design too that were used. So will the did the mixing kind of improve your results in the pipeline, or that uh
Yeah, but it depends on like what you want to optimize. So like if you always
okay.
like want to generate a binder that bind to your like certain interface of the target, I think yeah,
Mm-hmm.
definitely conditioning. Oh, this conditioning means like adding a diffusion potential. So throughout the diffusion process, like maybe like you can add more potential to that hotspot like to make the binder to bind to that, but also like you can additionally add the contact laws like during the performer process, like so here if you know oh this residue need to interact with this residue in the binder, maybe you can like upweight the loss on those residues. Does it make sense?
Hmm.
Mhm.
Yeah, yeah, yeah. That we can. Okay,
Yep.
thanks. Cool.
Mhm. Okay, any more question here on the bulls design part? Okay, then I can go through this the second part of this presentation, which is Protein Hunter, and maybe after that, yeah, we can get more questions. Oh yeah, oh, sorry. This Yep. So now I'm gonna like talk about the Protein Hunter, which is a paper we presented like probably like 2 or 3 weeks ago. And yeah, this is also relying on the structure prediction model, like AlphaFold 3, like structure prediction model without any fine tuning. And this is like basically based on like several like observation from the diffusion hallucination phenomena from this AlphaFold 3 STM model. So I think you guys already read the AlphaFold 3 paper and like some tweets about this paper and discussion about this paper, and then like there's some superior structure order and disorder regions like observed in AlphaFold 3 model. So for example, people like try to compare the AlphaFold 2 model and the AlphaFold 3 model on like multiple different targets, and this is an example of the disorder protein. And so when you give this protein probably the correct structure, it would be like there is some alteration and the other part is like highly disorder, so making this spaghetti-like structure around the protein because this is this disorder protein. But when you exactly give the same information, MSA, and the sequence to the AlphaFold 3, now you can see whole sequences are like folding into this 3D, the helical structure, bundle, and sheet structures instead of like generating the spaghetti structures. So people figure out, like AlphaFold 3 model, which use the diffusion model, like which is mostly so far used for the generative model purpose, are actually hallucinating more on disorder proteins, like compared to AlphaFold 2. So this model, AlphaFold 3, they explicitly fine-tune this model on the disorder proteins predicted from the AlphaFold 2 to reduce the hallucination effect. But if I remember correctly, like so far, the Chi model and the Wolf's model, I haven't actually fine-tuned on this disorder protein yet. And also, this is a tweet poster. Yeah, several months ago, I found this person was showing the structure of this BRCA2 protein. So as you can see in the left, this protein, like if it is existing alone, like it's usually disorder protein with like some helical like the order regions. So after AlphaFold 3 is fine-tuned the AlphaFold 2 disorder protein, now if you make a prediction of this protein, you can see this like spaghetti-like structure with a very low confidence score. But what if like you give any binding partner to this protein? So yeah, like this person give like two different proteins into the AlphaFold 3, which is BRCA2 and the red 51 protein, and then now BRCA2 protein like started to form this like helical bundles around this red 51 proteins, and actually folding into these like helical structures. And so we actually figure out that AlphaFold 3 hallucination when it is uncertain about this like prediction and the binding interactions. So this is actually not that great for the structure prediction purpose, but we thought that, oh, it will be actually really cool for the protein design purpose, because like if we can generate like somewhat plausible structure and then good looking structure, maybe we can redesign a sequence that can actually fold into that structure and like use it as a binder. So we actually use this adversarial like structure prediction models like default, like just for designing purposes. So do you guys have any guess about like what if like we predict the structure of this a protein of your interest and just random georepids of the proteins and like predict with the AlphaFold 3 like structure predictor? So yeah, if you don't have any answer, I can just show you this the result. So it looks like this. So actually this random G repeats, which is actually doesn't really make sense, are folded into the somewhat like okay structure, and it actually binds near to the protein of your target, right? So we thought that, oh, maybe model also like hallucinate on random like amino acid sequences, which are the out of distribution of the training data set. So the model never send this like sequences, just a random amino acid. But like since this diffusion-based model, like which is used for the generative purposes, they are and then AlphaFold 3 models are always like trained on this highly confident structure and the sequence, never trained on this adversarial sequence, like can actually try to generate a good looking structure as an output.
So what does it look like if you do this in absence of the target?
Absence of the target, yeah. So unconditional generation, it actually folds into the structure. Well,
if you only have the GGGG,
yep.
it folds into a structure and not just a helix.
So it depends. Sometimes, like, yeah, it depends on the random seed and it depends on the conditions. But in most cases, as you can see in this figure, yeah, some of the protein, like the all protein sequence, proline sequence, just fold into this like random helical bundle, random like spaghetti-like structure. But most of the structure sequences here I've shown, like they're folded into like at least like some
Ah,
kind of a secondary structures.
do you know about proline-rich extended helices?
Mm. Yeah, I heard of
These
it,
are,
but yeah.
yeah, we actually have, I just know about them because Bill DeGrado has some, in our paper, Bastian, we have that as a target, like a
Mhm,
little peptides called indolecidin.
mhm.
That is a proline-rich or proline-2 type structure, which I never heard of before. And
Got it.
yeah, I didn't know this type of secondary structure.
Mhm.
But yeah, now that's probably why it looks like this in the pure proline thing.
Yeah. And I think, yeah,
Um,
like if you look at the distribution of like existing PDB or cat data set, a lot of like proline residues are located in the loop regions of the protein. So maybe they're like very enriched in the loop regions of proteins. Yep. So yeah, we're like just wondering, oh, like what if we generate the random amino acid with this single type of amino acid, just make it like alanine repeats, arginine repeats, asparagine repeats, so, and like give it as a binder sequence and predict with the target. And then like we could observe that, oh, most of them are folded into pretty good looking structure, and most of them are like bind near to target. So you can definitely see some of them are like floating around the target, but yeah, many of them are like still ported into some structures and like bind to the target. And more interestingly, they're not just only like folding into the alpha helical structure. Some of them are like folded into the beta sheet structure, and some of them are like alpha and the beta mix. And I think, yeah, they are usually following the distribution of the this residue in the real proteins. So for example, the valine, these proteins are highly enriched in the beta sheet structures, and if you fold it with a valine repeat protein, it actually just folds into multiple beta sheet structures. So we could, yeah, probably think it as, oh, maybe modal hallucinate, but still like follow the amino acid features like throughout this predicting process. But the cool thing we figure out is these all AlphaFold 3 style models are trained with 20 different amino acid, but additional X token, which is actually the unknown token. But we actually figure out the unknown token really works well, and then we can really easily hallucinate with this token. This token is used as a token when in the like training the PDB training data set, if there are like the residue that are unresolved or like sequence uh, unknown unknown sequence, they replace everything into this X unknown token. And also for the known canonical amino acid or the amino acid with a different side chains, not the 20 amino acid, this 20 amino acid, they just replace everything with the X tokens. So we assume that, oh, maybe throughout this training process, the model learned X is like somewhat the protein-like token, but they haven't explicitly learned about the certain amino acid specific feature. So it has a like the minimum bias toward the specific feature of the amino acid, but still can fold into good looking structure. So yeah, we just like play around with this X token and like try to see whether we can like generate the binder or like just single monomer protein or the protein that binds to small molecule, etc. Everything like that the works that are similar to Bose design. So yeah, this is the video like showing that given a PD-1 target, you can just give an X repeat of the protein. So this pink part is a binder and this purple part is the target. But if you give it as an input to this structure prediction model, like this is a bolts prediction, then at the end, you can see this X tokens is folded into the well-organized structure and bind near to this PD-1 target. And it doesn't require any backpropagation or like reprediction. You can like just use the one single forward prediction of the structure prediction model. So it could be done in like 10 seconds. So it's very very fast and high throughput method. So if you do this process, just giving all X as a binder sequence and predict the structure, and then redesign everything with the protein MPNN, the soluble MPNN, and refold everything with the AlphaFold 3. And if you check the IPTM score, that they don't really look good. And we thought, oh, it makes sense because like although it generate like somewhat possible structures, it's not like super optimized and it's still like suboptimal solutions. And if you give it to the protein MPNN model, like since they're like just trained on the good looking structures, they will probably not gonna like generate the good sequences. And so what we have thought about is, oh, actually, but this is like still good as an initial starting point. What if like we can like just optimize the structure and the sequence more similar to the previous approaches? So this AlphaFold 2 cyclic paper, which was published last year from Chris and Sergey, is actually the paper showing that they can improve the structure and the sequence and the designability of the structure and the sequence by repeating the sequence designing and the structure prediction pipeline. So the goal here, they want to like design a very large protein that can fold into the design structure. But if you many like generated models, like they're like suffered if you increase the length of the protein. So if you like go up to like thousands of the residues, it really like struggle a lot. If you redesign with the MPN and refold it, it never like fold into that structure. So it's very hard problem. But they actually get initial design from this chroma diffusion model and the initial sequences from the chroma design pipeline and give it to the model and make the prediction using AlphaFold 2 and to get the per residue level of the confidence from the histogram contact loss. And they redesign the residue that have a really low confidence with the protein MPNN. So by mixing the losses from the protein MPNN and the initial like chroma design sequence and biasing upweighting the protein MPNN losses for residue that have a low confidence, they do this resampling method again and again and predict the structure again and again until it converges. So they repeat this process like in default like 10 different times and get a finer structures and redesign everything with the soluble MPNN and refold with the ESM fold model, which is like not the AlphaFold 2 model. They kept the ESM fold model for cross validation model. And after you repeat this, the cool thing is happening because like as you can see in the top part, uh the gray structure is a original chroma designs and these a colored structure are a structure coming out from the AF2 cycler after like multiple redesigning the structure prediction. But they don't look that different, right? They looks almost similar, but like slightly different. The secondary structure and the slightly different like distances between the helical bundles and then like sheets and the loop structure. But if you do this process, like you can really maximize the designability score, which is measured by the TM score. So what I meant here is like your finer structure and then your refolded structure from the cross-validated model. Oh, question.
Yes, I wanted to get an overview of how much more you were planning to present because I have some questions that I really would love to discuss with you, and yeah, wanted to see how much time we have.
Yeah, I have probably, yeah, I can go real fast, like later part, so I can probably done in 10 minutes.
Okay.
Mhm.
I'm afraid that many people might drop in 10 minutes.
Oh, yeah, yeah.
What do you
Okay.
think about the content? How we should structure it? I feel like is the what would you say is the meat of your presentation,
Mhm.
right? We already talked a lot about like this fact, just that like this is to me the most fascinating thing about Protein Hunter, just what happens inside of these models. What
Mhm.
yeah, just the surprise that you get a structure prediction out of some complete jumble sequence, and
Yeah.
then you can do your cycling stuff.
Yeah.
So, beside that meat, what do you think are the other good pieces we should go through? And then let's maybe focus on those.
Yeah. let's do it. Then I think, yeah, it's
Okay.
better to just like skip this overview about it. Yeah, so yeah, as Hannes introduced, we are like, we figure out like with a random like amino acid sequence, the model can hallucinate and generate a good looking structure. And by repeating this process, we can increase the confidence score rate and then the success rate of this design. So this is the whole like pipeline, the iterative sequence design and structure prediction. And the one observation we figure out, and we thought this is really cool, is I didn't mention here, but this light color on the leftmost part of this H target box is this the redesign the alanine count of redesign sequence from the cycle 0, and like this dark color represent the design from the cycle like 5. So if you get the first sequence from the first structure that all from X repeats, all of the design really have a high alanine content, because like in this model, the Boltz model, you use the same backbone atoms as alanine for X. So it has a like C alpha, and O, and the C beta. So if you redesign everything with the protein, man, they thought, oh, maybe like those residues are like just alanine. So just put the alanine on the residues. So like many of them have a really high alanine, but if you repeat this process, like you can actually decrease the alanine contents to make your designs to have a more high specificity
Okay.
by more different side chain. Yeah. Mm-hmm.
So do I understand right that in the beginning, sorry, what sequence do you start with? All unknowns?
Yeah, or all unknown for the default setting.
Okay. And then the model predicts helices mostly.
Yeah, mostly helices.
And then we think it's alanines. Everything is alanines,
Although
right?
as helices, I think like it's not always need to be a alanine, but I think because the backbone atoms and then maybe the distance between the side chains are not fully optimized, I think like this protein is just alanine based sequence.
Yeah. So if we have a very helical construct, then there's an alanine bias from the inverse folding model afterward. But then
Yep.
we refold,
Mhm.
and then we have a more reasonable looking sequence already, and then the alanine bias is less. And now here you're visualizing how this alanine bias goes down for several
Yep.
different targets over the steps.
True.
Okay.
Yeah, but there are like two different methods to reduce this bias as well. One is, yeah, we're gonna include in our code base, but one is like you can actually add the negative alanine bias during the inverse folding model sequence redesigning step. So in that case, like model like try not to place the alanine, but instead of like giving other residue to be sampled. But the second thing is it always like works with all X, but it's sometimes like better to mix with the other random amino acid. So now our sampling pipeline has a sample the sequence not only from X, but I mix 50% of the X with the 50% of the random amino acid, and you give it as an input. Then you can like sample the more diverse secondary structure. So it's also like just random thing, but if you give a random amount of sequence, the model also works. So we figure out, and then if you mix with the X, like it's more stable. So maybe like 50% of the X and the 50% of the random amino acid, and like generate the one sequence. Like, yeah, it could be a good way to generate the increase more diversity. And then in terms of like sequence and the structure as well. Oh, I have few slides, but this is mostly about the success rate of our unconditional generation and the conditional generation. So do you think it's better to discuss like some other stuff, or like can I
Actually,
just
a
stuff?
question that I would like to ask you would answer whether we should discuss it. Because how much do you think that we should be paying attention to these designability scores for evaluation? Like we always look at the diversity of the stuff that we generate, and
Mhm.
then we look at what is our diversity below the certain thresholds of like refolding RMSD. Do you
Mhm.
think there's value to that, so to say? There's yeah, is that the right way
Yeah,
to choose our
I
modeling objectives?
Yeah, I definitely think like maximizing the diversity, but also the success rate is the best way, and then like maybe yeah, our goal for the future. But I also think it depends on the target. Like some protein target like prefer like some like the helical like a structure as a like binder, but like some targets prefer more like beta sheets as a binder. And we don't really have a good understanding about that yet. So I think it also depends on the targets, but I definitely agree that like increasing the diversity is also a key question, like because by increasing the diversity instead of like just randomly designing the helical bundles, like we can improve the specificity of this protein. So it depends,
No, but I
but
mean,
Mhm.
even just, mm, yeah, I don't know.
Mhm.
I just want to try to get away from us as a community, everyone optimizing toward having higher
Oh, I think there is some connection. Oh,
IPM and highs after folding with um I cut out them.
oh, sorry. Yeah, the connection was not great. So can you repeat it again?
Yep, I'm not sure if I can repeat, so I would say just go ahead and ignore me. Um, my
Okay.
internet's not the best.
Yeah.
So I had a query regarding sparse autoencoders. Like maybe when you finish your presentation, we can talk about that.
Mm-hmm.
Yeah, sure.
Yep.
Please go ahead now.
Yeah, okay. Yep. So I'm gonna like just briefly show the result. So by repeating the cycling process for unconditional generation, as I mentioned, the problem of this generation model is like it really having a hard time for generating the large proteins. But by repeating this, we can increase the foldability of this like larger domains of the proteins and also the self-consistency of these designs. And even for the binders, we could achieve the like higher the confidence score for the interface and both like backbone structure itself. So it's helpful to for the binder as well. And by doing so, the protein enters after 5 cycles, even though like at the cycle 0, it shows a high the success rate. Now we can like push the success rate like to be higher. So here, like we could achieve the highest success rate with the both two model with the protein hunter setting and then like followed by the chai one. And then I figure out the AlphaFold 3 are less hallucinating than the others because I assume that these models like purposely fine-tune on the disorder proteins and like some Frankie designs. So maybe like the model already learned not to hallucinate and like generate, which means it's generating more of like the order regions like for the random and inconfident sequence. So I think yeah, my assumption is like since the model like, is fine-tuned on the bad structure and the bad structures like they're less hallucinating than the others. So we end up like using this two different models for our designing pipelines, but we are like try to expand into other like AlphaFold 3 styles models such as Rosetta Fold and also the Protein X. Yep. So we could also design a cyclic peptide, and cyclic peptide could be like done by just cyclizing the positional encoding of the proteins. So we could generate like different length of the protein given a this MDM2 target, but we are like try to expand this more to more difficult targets and like by collaborating with the Garov and the Griffin from IPD. Yep. For the small molecules, we also evaluated on these 4 different targets, and yeah, Protein Hunter could achieve the highest the FFO3 success rate measured by PAE, RMSE, and the PTM score. But yeah, as Hennis mentioned that like if you predict everything with the all X, the problem we often get is folding into highly helical structure because like as you know, most of the structure database are like having like a lot of like helical structure compared to the beta shed. So a lot of like generating models are like just generating the helical structure if you don't like specifically giving a bias or the fold information to the model itself. So our model, our pipeline also sampled most of the helical structure like during this structure all structure predictions tab. So the one thing we focus is, how can we actually maximize the structure diversity? How can we sample more with beta sheds? And the simple thing we have tried is we first started to just add the diffusion potential within the diffusion model, and then it sometimes really doesn't work because like we mostly focus on the angle of the backbone, so angle of the alpha helices versus the beta sheds. But if you give like that bias within the denoising tabs, like I often just like sample the multiple of like this lupulation of the proteins. So I figure out, oh, maybe it's not as simple as I thought. So but these diffusion models like get the pair features like as an input from the performer prediction. So I thought, oh, what if like we like steer this like prior of the pair features, like which is like given as an input to the model. So what if like we just kill the helical like signal from the performer itself? So for example, like as I mentioned before, most of the helical bundles like has the highest contact signal at position I and I+4, because like you're making helical bundles and like the positions here are making a contact with the position I+4, yeah, by forming these helical bundles. So we just purposely penalize the position I+4 by randomly just flipping the value of the hidden representation of the performer at the end of like these recycling steps. So after you do that, as I increase the helical bias more and more by increasing the negative helical bias constant, I could sample a more beta shift structure instead of like us steering the diffusion model like by adding the bias to the performer pair features. Like we could achieve the more diverse the structure, like, given a like same all X sequences. So I thought, which is, yeah, it could be pretty cool, like if we can like design the binders, yeah, towards small molecule protein, like, and also like giving this negative helix bias as an input to the model. And then, yeah, as you can see in this bottom, like when I add more helical bias, yeah, I could achieve like this alpha beta mixed proteins, like for this small molecule binder with also with the high success rate. So, yep, as you can see here in this histogram, the density plot, as I'm giving a higher bias, like you can see the sheet context is increasing. So, which is, yeah, pretty well correlated, and with this random, like just very easy way of penalizing the helical bundles. So yeah, just briefly summarizing about the application, we haven't externally showed this yet in the web lab, but we actually really want to like focus on these design problems. Like, so the first one is the multiple binder design, because the biggest problem from the backpropagation method is since you need to backpropagate, it requires a lot of like computational time and resources, and you usually repeat it like for multiple times, like more than 100 times. So it's really a long process. So it's really hard to work with this multimer protein. So people usually often like just crop the protein or like give the single like copy of this protein and generate the binder. But most of the times, if you do that, like sometimes this binder just bind within the interface of these two protein copies and like inner side of the protein stack, and it's hard to handle it because like those homo-oligomers already have a highly like defined like hydrophages that where the model like just think, oh, you should like give this binder to this the homologomer interfaces, something like that. So, but in this pipeline, we don't really use any backpropagation. This is just a one-shot forward prediction. So we can like just give it 3 copies, 4 copies of 5 copies of targets and just make it like single prediction. And you can like choose this target that can well bind to the outer part of this oligomers. And the other thing is we also really wanted to do the multi-motif scaffolding. And since both two trimodal like support the template information, you can just give a multiple different motif regions as a template and also the inter part of this motif as a template information, so the model can consider relative like position of different motifs. So now if you give this motif information template and the all X with between this motif and then on the N-terminus and C-terminus part, while you're like keeping this relative positions, you can redesign the connections and the N-terminus, C-terminus of these proteins. So yeah, we also want to expand this approach to multiple like different motifs, more than 2 motifs. And finally, yeah, we can also do the partial redesign, like especially for the antibody, if you want to, yeah, just fix the framework regions while like redesigning the CDR regions. Like you can give it all access to the CDRs and get the structure, redesign everything with the AGM-PNN or other like IGLM based method, and yeah, redo the cycle again and again. Yep. So yeah, this was a brief overview about my the current project, and then I think yeah, there should be definitely more room to improve. And yeah, as Hennes mentioned, yeah, I actually really want to focus on like designing more diverse structure and like solving hard problems. But yeah, this is like TBD, and yeah, I want to just acknowledge my collaborators, the Martin at Zurich University tutor from Adaptive Bio who suggested us like to do some experiments, and Sergey Garov and Griffin from the IPD. They do mostly the mini binders or the cyclic peptide stuff, and all the people from Solo. So happy to get more questions, and happy to do more discussion as well.
Yes. And then thank you, Yehlin. And I would say the same thanks to Adaptive Bio. That's a really design to labs that do not have much wet lab capability, I suppose. And yeah, the question I would ask is, do you have an intuition on inverse folding? Mayank, maybe also mute yourself while, because I think there's a little background noise. And yeah, for design soluble MPNN, for example, and other inverse folding models, how important do you think inverse folding is? Should we be inverse folding the interface after we designed? And is there like considerations between soluble MPNN and protein MPNN besides solubility? Yeah, any thoughts around that?
Great. Yeah, I'm not like sure about the other inverse folding based method, but sometimes I heard that like from mostly from the experimentalists, if they really have the unique form of the target and binder, the protein MPNN kind of like inverse folding models are like try to make a like simplest as possible interface. So sometimes those models are not really generating the interface that are really having a high specificity toward one single target. So they can like multi-specific or like having also like cross like side effect with the other like proteins, which is like not on the purpose. So yeah, I think maybe like it's good to be like focused on how can we actually maximize the specificity. Like specificity can be achieved by hydrogen bonds and like the multiple different of like interactions between the residue different interfaces. So yeah, my just like thought is maybe like since this inverse folding models are like not perfect, there should be also room to improve like in terms of specificity. But I think I definitely think it's a great tool, and then like you can really maximize the solubility like by using the soluble MPNN or other tools. So yep, that's my just intuition. But
Okay. Yeah, these inverse folding models are really doing some magic. I will probably the recipe. Or what do you think is the reason why some other inverse folding models have this issue with like generating very simple interfaces?
Yeah, I think the first of all is like the structured data set itself is like limited. So I talked with Martin and people before, and then the reason they think like inverting the structure prediction model like can sometimes do better at like designing the interface is maybe like is the model informed like much more like much diverse like sequence database like through the MSA, the sequence and structure version alphafold database. So more of like a different diverse the structure information like than the simple like PDB based like structure inverse folding model. So I'm not like totally sure about this, but maybe then like the diversity of data set could be the one reason. And yeah.
Yeah.
Mhm.
Okay. Any other questions?
So like I had a query regarding the sparse autoencoders that
Mhm.
can be used to expand the structural diversity. So like we can use those representations
Mhm.
they I think so learn a compressed latent manifold of
Mhm.
protein structures. So plus sparsity can be a very good combination. So what are your thoughts on this?
Yeah, definitely. Yeah, in our discussion, we also wrote, oh, what if like we just use this person autoencoder like on our pair last pair feature from coming out from the performer and then like retrain it, and then maybe we could guess some like parameters that are activated like for the just helical like structures and purposely kill the signal and regenerate the representation or something like that. And yeah, there that is still in our like to-do list, and I'm trying to do that. But like one concern is what if like just modal like simply just like kill the position and I+4, then is it just like simple just as simple as like just killing the that the pairwise representation from the like performer. So I'm not sure about the research, but yeah, that's a very good direction.
Cool, thanks.
Yeah, but I think definitely if you can figure out the better way to generate a diverse performer, maybe you can like better generate the better structure from the diffusion model.
Yeah, I totally agree. Plus, like, there are certain pitfalls too with the sparse auto encoders and stuff, like the mode collapse and the
Mhm.
latent channels that are not like interpretable.
Mhm.
Like those issues are there, but I think so it can be a very good research direction. And like for the next part, I think so you can go here with that.
Yep. Thank you. That's good.
All right. Yehlin, any other things you think are worth sharing?
Uh, oh, there are some questions from the chat room, so maybe I can read them.
Yeah.
Yeah, really.
Maybe the.
Mhm.
Okay, go ahead.
Yeah. Different combos pair of repeating just one many
So
times.
essentially, the question is, right? You have your AAA, but did you also try half A, half proline or so?
Yeah, I think definitely it will be helpful. And then I definitely think if you, for example, like the maximize the amino acid residue, they're like highly appeared in the beta sheets, maybe you can like better to form that beta sheet like structures. And yeah, definitely you can increase the diversity. So in our code, we're gonna release in this week, and we have two different you can as an input, you can like specify, oh, whether you want to like generate everything with the X, but randomly sample the sequence from the like random amino acid distribution. So yeah, definitely, we checked that for unconditional generation by randomly mixing the amino acid, like can maximize the structure diversity because all X are like try to just generate the simple sequence structure and sequences. Yep. And what is the difference versus increasing the number of diffusion steps? Uh, I think yeah, I haven't tried for all of different targets, but I usually use the 200 different diffusion steps, and then for some targets, for example, like for the nucleic acid binder, I often get a like problem to get a good structure, so I increase like 200 to 400 diffusion steps, and it increased the success rate a little bit, but one thing I figure out is like in the diffusion model, like as you at the very early stage, yeah, it mostly like decide the fold and the like global like dock structure, and at the very later part, like it's like locally changing the things. So I think if you want to like increase the diversity, you need to like focus more on the earlier step of the diffusion steps. But I haven't explicitly tried to like yeah compare that.
Okay.
Oh,
Then, Yehlin, thank you. I will call it a day here, and everyone else, thanks for joining too. And yeah, maybe see you. Oh, maybe I should shamelessly advertise that this Thursday And I wanted to ask you, or it comes to my mind, Yehlin, now, because I would have asked you if you're joining, and maybe we can chat more then.
Yep. Sounds good.
All right. Yeah, this Thursday, 5 p.m. at MIT. Cool.
Mhm.
But then see everyone around. See you next week, guys. And have a
Yep.
nice Monday.
Thank
Thanks,
you. Yep.
Yehlin. Super interesting.